Okay. So today's material is covered on the exam on Wednesday. It's kind of finishing out what we've already covered. It's basically just antibodies detail. All right? So you would expect questions all the way from the first day of class until today, through today's lecture. Is with me? 50 questions, right? Multiple guess, or choice, depending on how much time you put in, I guess. Let's see. I think an advantage or a way to study that's an advantage is really to focus on each lecture and maybe try to identify, okay, what are some of the key take-home points of that lecture? Hey, what was the lecture about? Right, so if it's an inflammatory lecture on inflammation, then maybe cover a couple of the key topics within inflammation and then start working through details. Make sense? If you can explain every slide in the inflammation lecture, you should be fine. How detailed do you need to go into? Well, I would first go through the whole slides lecture on inflammation and make sure that there isn't something you're like, gosh, I don't remember talking about that. Like, if you didn't remember talking about it, you probably should go back to the YouTube lecture and find that spot in the lecture and maybe just listen to that 5 minutes and say, oh, okay, now I remember. Okay? All right, at the end, we'll have a couple of review questions, and then we can talk a little bit more specifically about different topics and kind of go back and forth, okay? But first, we've got to finish up the material. Okay, what do you get when you cross? Oh, by the way, these are not on the exam. Okay, I had an email about that. Like, is there any possibility you're going to ask any of these riddles on the exam as extra credit? I'm like, no. If you can explain what you get when you cross a caterpillar and a parrot, I don't care, okay? But it's fun, so let's hear it. What is it? Caterpillar and a parrot, what do you get? A walkie-talkie. Why is Cinderella bad at basketball? Why is Cinderella bad at basketball? What's that? She only has one shoe, it's a glass slipper, so that's probably the real answer, but that's not what they wrote down. She runs away from the ball. Not to mention that she's running around with a glass slipper and only one at that. What do you get when you cross a tornado and a cow? What's that? Alabama. Don't go there. There's probably somebody from Alabama here, and I feel bad. Okay, so we're not. But then we can just switch over to like Mississippi or something. Right? Milkshake, tornado and cow. All right. Alabama's pretty good. I mean, it's football season, so. Okay, so we're picking up here, talking about antibodies. I wanted you guys to take a look at these lectures. I want you always to take a look at these lectures, but particularly today because there's a lot of detailed drawing that I'm not going to put on the board because what I drew this one time, I tried doing it in subsequent semesters, and I'm a really bad artist, and so I kept this paper and like took a picture of it and then made it into a slide. And so it took me like hours to shade it, and you'll see here in a second if you haven't seen it yet. Some of you are like laughing, like, seriously, that took you hours? Looks like you had your kids do it. It probably would have looked better if I did it, but. Okay, so the different components of an antibody. Let's start with, an antibody is a protein. Okay, an antibody is a protein. It has two main regions. It's got an antigen binding region, which we call the FAB region. It's also the variable region. We'll get there. And then it's got a crystalline region, or the heavy chaining, or what's called the FC, constant region. And this picture will show up here in a second, and it'll make a little bit more sense. There are different types of valences with antibodies. Valences is a term, or a valence is a term in immunology that refers to the number antigen binding sites. The number of antigen binding sites that's possessed by one single antibody molecule. So if you have a dimer, right, which it looks like a Y, it's got 2 binding sites. There are 5 types of antibodies that we're going to look at. Okay, so just hold the phone as far as what we're going to look at. It's on the next slide. You can write it here for sake of completeness. So IgE, okay, IgA, IgD, IgG, and IgM. Okay, so you have A, D, E, G, and M. If you go alphabetically. A, D, E, G, and M. So some characteristics that you see on this particular slide, on the left slide, is this concept of antibody secretion. What cells are manufacturing antibodies and secreting? B cells and what else? And plasma cells. Okay? And they can undergo, these antibodies can undergo what's called class switching. So that means that the B cell that makes the antibody modifies from like an IgA to an IgD, or an IgA to an IgG. So it switches to a different class. And when it does that, usually it's swapping out the variable region, and it keeps the constant or the heavy chain the same. There might be certain circumstances where that's valuable. And you'll see here in a minute when we characterize the different categories of antibodies. That B cell has the ability to go through an alteration. It can adapt, right? It can adapt through a class switch and start producing a different style or type of antibody from the same B cell. Okay, so that adaptation is taking place. That B cell is able to respond or evolve even very quickly to making different types of antibodies depending upon what's necessary. This terminology, the third box down, of affinity maturation. So this is a term that means that the B cells themselves are producing antibodies that have an increase in affinity for the antigen during the course of an exposure or immune response. So as they make antibodies in the beginning of that exposure, they may not have as great of an affinity or ability to bind, but as sort of the exposure goes on, they increase their affinity, or affinity maturation is another name for it. And the last but not least on this particular slide is this concept of memory B cells. And we're going to talk a little bit more about those here in a minute, but they're a subtype of B cell, and that subtype of B cell is formed when you have a first infection and it subsides or goes away, and these B cells that were activated convert over to memory B cells. So the next time you get a response, you've got that particular B cell with the coding to make the antibody that was necessary in order to fight off that particular disease the first time. So you never get a memory B cell until after your first exposure. Make sense? And this is one of the concepts, or really the concept for vaccinations, is you can actually deliver a antigen that is defunct, non-functional. So you can take an antigen or a peptide sequence and you can boil it, you can denature it, you can change its three-dimensional conformation, and then you expose the individual to that, which it won't infect them because it's non-functional, but if it's close enough to cause the B cells to bind the antigen, then they will make antibodies towards that antigen. Does that make sense? Okay, it's the same concept for like the flu vaccine. You guys ever been vaccinated for the flu? How many of you have been flu vaccinated? Okay, so I'm only, hold your hands up. I'm going to look at the people that aren't raising their hands and stay away from you all. Okay, perfect. All right, got it. So if you read carefully on the flu vaccine, it'll say things like, we are not giving you a functional virus. Okay? So you cannot get the flu from the flu vaccine because it's a non-functional virus. They completely altered it. But what it will do is it'll trigger the response of the immune system to start creating defenses against it. Okay? Okay, we'll talk about memory T cells here in a minute, a little bit later. Okay. So the 5 types. So this is a very important table. There's a lot of information here. Well, let's go sequentially in alphabetical order. You can see this dimer that we talked about up top. So this IgA dimer. And there's 2 subtypes that we're focusing on. We're focusing on IgA1 and IgA2. You can see that these immunoglobulins, or these antibodies, that's what Ig stands for. It stands for immunoglobulin. So that's an antibody. And the IgA, or the immunoglobulin A antibody, is in the shape of a dimer. So let me just fast forward to this slide. Hopefully you've already looked at it. So this is a dimer, right? So you've got 2 Ys that are stuck together for the dimer. This is the IgA example. And I think I misspoke a little earlier. Let me say it again. If there's a dimer, how many binding sites are there? I think I was talking about a dimer, which is really a monomer. I said 2, right? Did you guys catch that? So a monomer has 2. A dimer actually has 4. So I think I misspoke. We can look back at the record, but let's correct me here. So if we go here to a dimer, a valence of 4, IgA, there's a high secretion of IgA in the blood. And there's a, high concentration of IgA in the blood, and high concentration of IgA2 in body secretions. That would be like sweat and saliva, okay? IgD, the monomer, the lower concentration in the blood, but it's found on the surface of B cells as a type of a receptor, so it's very commonly found. And it's been shown to activate these basophils and mast cells to produce antimicrobial activities. Another type of monomer, IgE. This is our allergy antibody, IgE. So after the exam, we'll go into more details on these particular types. But what you should probably know for this exam is it's in the lowest concentration in circulation, like in the bloodstream, of all the immunoglobulins or all the antibodies. But it's the one that facilitates or regulates an allergy response, IgE. It also is what's triggered in a parasitic response. So let's look at IgE, for example. So how many of you know you have an allergy? Like you have a food allergy or a drug allergy like penicillin, you can't take certain antibiotics, you get like a rash. Or maybe you have an allergy to like pollen or cats or dogs. Okay, so all of us that are raised in that, that's me too. I mean, most people are allergic to something, okay? My 10-year-old tells me she's allergic to school. And I tell my wife that I'm allergic to broccoli. But those 2 were not necessarily acceptable in my house. So this allergy response that we have is the IgE is released, and it acts on mast cells, and they release buckets of histamine. And so think about if you're going to have during springtime, allergies are high, and we take what kind of medication that's an over-the-counter? An antihistamine, okay? So are we blocking IgE from being produced? No. You're still producing the IgE. It's just their activity on mast cells to make histamine is blocked because of the antihistamine that you took, okay? All right, IgG. This is the most common, 80 to 85%. Okay? 80 to 85% of the circulating immunoglobulin, so it's in the highest concentration, another monomer. And then IgD, excuse me, don't mean to skip over this part. This one is the one that is transferred, one of the very important immunoglobulins that's transferred to the baby from a breastfeeding mother. It also is delivered to the baby in utero as it readily crosses the placenta. So mothers deliver some of the adaptive immune system to their offspring. okay, in utero, in the womb, and then afterwards while they're nursing. And then IgM, which is the largest in size, it's pentamer, so it's got 5 of those stuck together. And it's secreted in the first exposure, the primary immune response, the first time you see the antigen. Okay, and then here's the drawing, the famous drawing that I've been talking about. So there's a lot of hype, but really it's really not that complicated. But you can, if you look very carefully, you can see that I actually had them get in pencil, and then I went over it in colored ink as I kept messing up. So, what is shown here? What type of formation? Monomer. Very good. So you can see this is the antigen binding site, which is the combination of these 2 prongs. You've got a light chain and you have a heavy chain, and you have that repeating, a light chain and a heavy chain. You can see the variable portion is the portion that makes up the antigen binding site. And then the constant portion is also known as this heavy chain, and it is not altering. So we do class switching, it's this constant that actually stays the same, and you swap out the variable region. Does that make sense? And then you can see the nomenclature from that first slide that we started on. Here's our FAB, also known as our antigen binding region, which is the component of these 2 prongs, which is representative of this. And then here's the crystalline-like region, which is this, portion which is down here. Okay, make sense? So you can see that, again, even with the monomer, this is always why I get confused, even with the monomer, how many binding sites are there? There's 2. But you would think mono meaning singular, right? So you've got a single antibody that has 2 binding sites. So we would call that duplication what? We'll talk about this. I talk about, what's that? Redundancy. So here it is in the simple architecture of a single antibody molecule that's in its simplest form, and that's a monomer. It actually has 2 binding sites. So it's not just enough for one antigen to be able to bind to it. You have the ability for multiple antigens to bind, so it could actually be triggered. Yeah.
And both of those sites, can they only bind like one
Yeah.
antigen?
They will only bind one antigen, but you could have one on both sides at the same time, or you could just have one on one. Okay, so the different formats, and so we've looked at this a couple of times. I flashed forward on Monday to this. The monomer is sort of our classic antibody figure. So anytime you're reading in a text or you see pictures of an antibody, they never draw the pentamer. They always draw the monomer. You never even see really the dimer. And you can see the different configurations, a lot of similarity, a lot of redundancy in architecture, so we're saving ourselves some energy biologically in manufacturing these different antibodies. But you can also see how you could do some class switching relatively simply. Like you could move from an IgA over to an IgE relatively easy by just making that antigen binding domain different as you manufacture it from a B cell. Okay, so we're going to transition at this point into some of the mechanisms associated with B cells. And then we're going to wrap up towards the end of recapitulating what we talked about on Monday with T cells and showing you the steps of T cell activation, and that's how we'll end the lecture as we end on memory T cells. Okay, so B cell mechanism of action, and then we're going to go back to T cells, which is what we started with on Monday, and that's where we'll finish up. Okay, we should have a little time to look at a couple of questions, examples, take some questions, maybe talk a little bit about the exam, okay, comfortably kind of roll into it. It wasn't a heavy packed lecture because the exam's on Wednesday, and that was intentional. So autoreactive B cells. We talked about autoreactive T cells. Who can share with me what happens with autoreactive T cells? Yeah, just what happens with a T cell that's autoreactive? Do you let it continue if everything goes well?
Break it down in the lymph, or if it leaks out, it has 3 pathways to self-destruct.
Okay. So you'll break it down in the lymph node, which is considered what? What type of tolerance is that? Central tolerance, or in the thymus, right? You'll break it down in the thymus. is really where it is. Not lymph nodes, the thymus, that's central tolerance. If it makes it out of the thymus, it makes it to a lymph node, or it makes it somewhere else in the body, then you have three pathways, which is not called central tolerance if it's outside of the thymus. It's called what? Peripheral tolerance. Who can take a stab at the three different ways to battle an autoreactive T cell that's made it out of the thymus? One had a really cool name, energy. Another one had another term, apoptosis. And then what was the third one? A suppressor T cell or a regulatory T cell. Very good. And about how many T cells do we make that we destroy? What percent are destroyed? Huh? About 95% are signaled for apoptosis or destruction. Okay, here with B, that was very good, by the way. Here with B cells, so I agree, some of you are ready to take the exam. It's about 90% apoptosis instead of 95. A little less, but about the same kind of statistic. And the autoreactive B cells only go through a process known as central tolerance, and that's not in the thymus like with T cells. What tissue do you think it's in? In the bone marrow. Very good. So central tolerance for B cells happens in the bone marrow. Same deal. Is if they're autoreactive, you signal them for apoptosis. There is a reprogramming that is attempted, okay, in the bone marrow, because they're in a younger state. Remember, pro-T cells come from the bone marrow and go to the thymus, and that's where they become immunocompetent. So B cells are going through central tolerance in the bone marrow, so they're even earlier in their development, and so there's the opportunity to try to reprogram them. Where with T cells, you don't reprogram them, you just trigger them for apoptosis. So here there is an attempt to try to reprogram them, okay? This slide here is really important because I want to point out the role of the helper T cell. So it's kind of got this unique role. A helper T cell has what CD marker on it? CD4 positive, right? These are CD4 positive T cells. What's the other type of T cell? CD8 positive T cell, which is also referred to as what? A cytotoxic T cell. Very good. So these helper T cells, there's a name for them in a lot of the literature. They're called this linchpin of the immune system. So they're a critical cell type because of the crossover that they can do. Do you guys remember that big diagram that we drew up here? Yeah, Monday? I'm trying to figure out what day it was. Monday, right? And so we talked about on the left side of the chart or of the board, we talked about our first and second lines of defense, our innate immunity, and then over here, kind of in the middle, we talked about our adaptive immune system, right? And we broke adaptive into B and T cells. Well, over here where T cells were, the helper T cells, they had pathways to go help out with inflammation, right, with macrophages. They have pathways to help out with cytotoxic T cells. And here they even are showing a pathway of stimulating a B cell that's naive, right, that's exposed to antigen or antibody, causing them to proliferate. And so these helper T cells have influence over macrophages, cytotoxic T cells, as well as B cells. So this linchpin cell type is very important. That's become more obvious of the clinical need for them and what happens if they go away when we start lecturing after the exam on HIV. Because I told you that these guys are the cells that HIV targets. Okay, and that's why the virus is so deadly. And you won't get HIV questions on the exam, but I'm just trying to connect the dots between this unit and next unit because we have to do an exam at some point. But so we're kind of straddling exam one with immunology, and this is a nice point to kind of pause and take an exam. But I want you guys to see that there's going to be a continuation that takes place. Okay. So how do antibodies combat antigens? They can do these in two different ways, either directly or indirectly. So with our direct response, we've got neutralization, agglutination, and precipitation. Okay, so neutralization, oh, this was a little premature, sorry. Neutralization is referring to the fact that it binds and blocks the activity. Binds and blocks the activity. Agglutination is you clump the insoluble antigen. What does that mean? Well, you got the antigen and it starts to clump together. Now it actually won't be able to be dispersed as readily around the body. Does that make sense? You're kind of gathering it together. And then precipitation means that if it's soluble, you bring it out of solution. So you make it precipitate out. And if it precipitates out and becomes a solid, it's harder for it to float around the body. Does that make sense? If it goes into solution and stays unprecipitated, then it can disperse throughout the entire body. So these are the ways antibodies act directly: neutralization, agglutination, and precipitation. Then there are some abilities for indirect effects, where the antibody is going to cause something else to act. Okay, so these direct effects, the antibody is responsible for directly. So think about precipitation. That's probably a more difficult concept to visualize. So if you've got an antibody that finds an antigen, that antigen is in solution, it's floating around. As soon as it binds to that antibody, now it forms a solid. And that solid can start to clump and group together. And we will see that after exam 1, okay? Precipitation is kind of a cool way for, instead of it flying through the solution of the blood, it actually now starts clumping, and then you can actually trap it and get rid of it. All right, so with indirect effects, so let's look at this figure. So this figure is related to indirect effects, where we've got opsonization and phagocytosis up top, and then we've got inflammation-mediated activity on panel B. So let's take a look at opsonization and phagocytosis. So opsonized cell complement is involved with this. The FC portion of the receptor is binding. You can see the phagocytic cell now is been triggered. Opsonization means that this has been primed for engulfment or phagocytosis, and then it chews it up. So with opsonization and phagocytosis, antibodies stimulate, say, a macrophage to come in and gobble up the antigen and get rid of it. This is indirect because the antibody's not doing it all by itself. And then with inflammation, same thing is you've got the recruitment of a neutrophil that's being brought to the area from the release of complement. And this neutrophil, or you could actually even write here neutrophil or macrophage. The neutrophil or the macrophage can utilize the oxygen metabolites, the reactive oxygen species, to actually destroy that antigen. But in so recruiting neutrophils or macrophages, you're stimulating a local inflammation. So local inflammation. And this reactive oxygen intermediates, or these oxygen metabolites, just remind me, what do we know about inflammation in that local tissue? If you have a wound bed that becomes infected, right, what are some observations? What does it look like? It's red, it's inflamed. What's that? Who said it over here? Hot? Somebody say hot? Yeah, it's warm to touch. So are those local activities, or are they systemic? They're very much local. Do you get hot all over? Do you run a fever? No. You're only warm where that infection is, right? Okay. So indirectly, utilizing another cell type, or directly, the antibody is having activity all by itself. Okay, memory B cells. So these memory B cells are quite fascinating, and we'll talk about memory T cells kind of at the end of the lecture here in a bit. So the primary response is mostly an IgM response to begin with. You guys remember the IgM? So the IgM is what? What kind of molecule is it? It's the big one, it's the pentamer, right? So the IgM is the largest in size. If you go back to that table slide, it's the largest in size, and it dominates the primary response, which is the first few days. So if I go back, what is he talking about right here? Secreted in the primary immune response, and it's the largest in size. It's this guy right here, and it's the first one on the scene with the response, followed by high levels of IgG. So followed by high levels of IgG. Now, the secondary response, the next time that you're exposed to it, the IgM levels are about the same, but IgG is 4 times higher than the initial response, and it's immediate. So let me give you an example. Let me give you an example about memory B cells. If you're doing an antibody, okay, to a virus that doesn't mutate all that often, okay, or maybe it doesn't mutate at all, your memory B cells are what are responsible for that immune protection for the future. Okay? So if you take like measles, for example, right, and you're exposed to it initially, and in some of these cases, because when we expose patients to an inactive antigen that's from like the measles virus, you might have to have multiple exposures because you're not giving the live virus. Okay, so you have to have, oh, they gave me a shot and I gotta come back and get another one. Like, what the heck's wrong? I mean, I know it went in, it hurt, the nurse was horrible, or whatever, you're a wimp, and so you cried, you don't wanna have a shot again, right? So this takes a little time. It takes 3 to 5 days, call it about, a half to a week long before you're going to start generating antibodies in your blood or have blood titer levels of IgG and IgM. But if you get a booster the next time, you start making those antibodies even more quickly. And this is the reason that you're now immune, is you've already seen it and you can fight it off. Okay? Now, I picked that one because there's a couple of things that can happen. Is there could be viruses that are very old viruses, and we were immunizing populations for a long period of time, and then we like to say that the disease is virtually eradicated. Right? That's a word where it's no longer in existence. Well, there are places where maybe there wasn't vaccines, or maybe the virus lay dormant, or maybe it mutated. Okay? And so that's why you might, for all of those reasons, you might hear of, well, why is this vaccine coming back? Right? Why are we having these problems again? So viruses are susceptible to mutation. There's no question about that. And this is also one of the concepts, as you can imagine, for like biological warfare. Right? So there are groups that are looking at how they can leverage knowledge about biology and mutation and try to cause major problems in certain population groups. Okay. Yes?
Is IgG the only one that a memory B cell will trigger, or can it also do IgD depending on what it is that it interacts with?
Yeah, no, so all of the B cells are capable of a memory B cell subset.
Okay.
Yeah. So I'm just giving you examples that are most common examples for the purposes of this class. But if you looked at specific diseases, they're going to use different types of memory B cells. So for example, we'll, after the first exam, we'll start talking a little bit more about the allergic response. It's the same thing for the allergic response. Unfortunately, the first time you're exposed to an allergen, it's not nearly as bad as the second time. Okay? So how many of you know you're allergic to bees? Anybody allergic to bees? Or peanut allergy? Okay, what's another more pretty common? Penicillin. Okay, so Or shellfish. I have a daughter who's allergic to shellfish. Are we allergic to shellfish? She loves like lobster, crab legs. She loves that kind of shellfish, but she's allergic to it. And we didn't know it until, so every Valentine's Day we do like this fancy dinner at home. And we all dress up, and my wife makes this big spread, and we usually have like, you know, surf and turf. And so like a couple of years in a row, she'd always get real red in the face and said that her throat itched. And then one year it was like really obvious. She's like, I don't know if I'm having trouble breathing. And so, you know, you gave her a Benadryl and everything was fine. But we were basically sensitizing my daughter for like 3 years, because we'd only really have, you know, that kind of food once a year, you know. And so that's the secondary response. And so those of you that have gotten stung by a bee, maybe it was a little bit more aggressive than what you normally would have expected. And the doctor said, you might be allergic to a bee sting, so you probably shouldn't get stung again. You got stung again, it was even worse. Okay, or maybe the same thing happened with like peanut butter or penicillin or shellfish. And so that's this happening, but with IgE. Okay, good question. All right, so now we're shifting back just a few slides to kind of refresh what we learned on Monday, put it in the context of summarizing as we kind of wind down the new material. And we're coming to the top of the hour. We'll be done here in about 5-10 minutes. Got a couple of review questions for you, and then I'm yours until class is over if you guys want to talk exam stuff and how to study. Okay? So, what occurs during T-cell clonal diversity in generation? So this all takes place, as we said, in the thymus. T-cells in the thymus. So they become immunocompetent. That's what this slide is actually saying. And there's a molecule known as thymopoietin that is released by the thymus to stimulate the maturation of these T-cells. And so you can kind of see from this cartoon, you've got precursor T-cells that come in from the bone marrow. So you can write in here, from the bone marrow. They get into this proliferative phase. This is the thymus. And then they go through apoptosis and screening. And of course, you've got a few of them that actually leave that are immunocompetent and they're non-self-reactive. And this whole process is stimulated by thymopoietin, central tolerance mechanism. We looked at this chart, but I've added some more detail because, in my opinion, over the years, showing this chart once isn't enough. So it's nice to come back to it because there's a lot going on this particular diagram. And I added in another cell that wasn't in the slide earlier, and that's the somatic cell. So a somatic cell just means any body cell, any nucleated body cell. And so a nucleated body cell is not typically an antigen-presenting cell. But we know that cells can get infected. Those cells that get infected can actually still present their antigens. So I don't want you guys to think that an infected cell doesn't do anything. An infected cell will actually display the antigen on its surface. But an antigen-presenting cell is the more commonly circulating cells that move around that are going to grab up antigens and display them. And so the pathway that we looked at is an antigen-presenting cell with an antigen, like a dendritic cell, for example. They can present to either CD4 positive T cells, the helper T cells, or they can present to CD8 positive T cells. They can go either direction. Now, the somatic cell, this cell presents only to CD8 positive T cells. And maybe what might be a little misleading about this slide is it looks like the somatic cell is entering the lymph circulation. So this is not to indicate that it's entering. This is to indicate that it utilizes this pathway. Because that cytotoxic T cell, where is it going to be found when it's circulating around? It's going to be all over the body, circulating the lymph, and then it goes out. So if it finds a somatic cell that's displaying the antigen, it's going to kill it. Make sense? So I want to make sure you understand that this diagram is to show, maybe I should put this somewhere over here to show that the T cells actually find it. Does that make sense? versus it entering into the lymph node. That might be better. So the dendritic cell presents either CD4 or CD8. The somatic cell that's been infected displays, and then a cytotoxic T cell is what it presents to, but the cytotoxic T cell has to go find it out in the circulation. You guys with me? Okay. So just to remind you, as this works through, this CD4 positive T cell presents, is presented the antigen by the antigen presenting cell. You've got effector cells that are manufactured, proliferate, they go into the circulation, and then they are distributed downstream, and out in the periphery, these T helper cells, these T helper cells, CD4 positive effector T cells, can trigger macrophages and inflammation to kind of do its thing and clean up the problem. Simultaneously, you can get the antigen presenting cell presenting to CD8 positive cytotoxic T cells. We talked over here about the activity that needs to happen, where you've got interleukin 2 being released, binding back to the T cell. So now this is an autocrine co-stimulation event, triggering the production of memory T cells and effector T cells. And these cytotoxic T cells then are the soldiers that go out. They find those somatic cells that have the display, right? They find those somatic cells that have the display, and out here in the circulation is where they kill them. Make sense? Okay. Questions on this slide?
Just real quick, you said that the cytotoxic T cells have to go find it, and aren't they kind of called out by the cytokines? So don't they kind of know where to go?
Yeah, so they're being recruited by chemotaxis or chemokinesis, where like a macrophage is trying to recruit other macrophages and lymphocytes to come into the area. So they're going to see that gradient and migrate in. But in order to find it, they first have to be dispersed. So they go into the lymphatics, and from the lymphatics they can migrate out into the vascular system, and they can migrate out from there. Okay. Okay, this is just what I said, a list of what I said, same type of picture. The activated T cells enter the circulation, right? The T cells migrate to the side of the antigen. They cause macrophages to be activated. These release inflammatory cytokines that recruit new macrophages and even neutrophils to the site of the area. Then the activated CD8 positive T cells and the cytotoxic T cells, they are entering the circulation, and they go kill the infected somatic cell that's displaying the antigen. Right? And then of course I mentioned a second time, don't forget that this co-stimulation event of IL-2 happens up here in the lymph node before the cytotoxic T cell gets out into the circulation. Yes, sir.
For the killing of the cell, is that an apoptosis or necrosis?
This one right here?
Yeah.
It would be necrosis.
Okay.
Yeah. So the cytotoxic T cell is going to lyse the cell. Actually, I think I have a slide on that, which is coming up. Yeah, this is the next slide. Okay. But before we go to that, so hold that question, I want to make sure you guys remember this nomenclature. So the dendritic cells are presenting to CD4 positive T cells, and this interaction is CD4 positive on an MHC class 2. Remember that? And this is a CD8 positive on an MHC class 1. And you remember those MHC classes are what tell you you're you. They're what tell your cells that they belong to you. That's the self signal. Make sense? So, for example, if we go back into here, self-reactive, if you generate lymphocytes, these are T lymphocytes, that react to your own MHC class, that's considered self-reactive. You guys with me? Okay. Just want to make sure that we're kind of reviewing as we go through to this complicated, detail. Okay. So, Richard's question: how do these guys do this? So, these guys, I think I misspoke. I told you it was necrosis. This is apoptosis. So these cytotoxic T cells, right, these CD8 positive T cells are going out and creating direct trauma to the cell, but the trauma that it creates as it lyses the cell triggers it to go through an apoptotic event. So we got two different mechanisms on cytolysis. So we're calling it cytolysis, where we're lysing the infected cell, but it doesn't degrade to a point where we get a necrotic response. It goes into a very programmed getting rid of the cell, okay? So I think that's why I said necrosis, because of the lysis, but it doesn't go all the way to the point of like where you've got traumatized tissue with fibrotic response, okay? So intrinsic pathway, these are just receptors. The intrinsic pathway is the damage to the phospholipid membrane causes FAS-L receptor to bind to FAS, and that causes an internal event to the nucleus to say, pack everything up and let's shut down the cell. And then the extrinsic pathway is you've got perforin and granzyme binding to caspases, and then the caspases signal intracellularly to trigger the nucleus to go through apoptosis. So you could almost view, we look classically at necrosis versus apoptosis, which is something that we should talk about here in a minute when we get done with the whole lecture. And then the activity of cytotoxic T cells, where we've got cytolysis, it's almost as if it's something that's halfway in between. Because you lyse the membrane, but you still go down to programmed cell death. Make sense? Questions on that content? All right, last new material, and then we'll talk about some review questions. We've got memory T cells. So these memory T cells, they've become experienced by prior antigen exposure. And in this situation, the second time, like with the B cell, it's a faster and a stronger immune response. And so if you look at the diagram below, we've got some unique details that describe these cells. So here's our memory T cell subsets. These are effector cells in this box where we've got a naive T cell, and it proliferates. It is stimulated with interleukin 2, and now it's an effector T cell. And you know, if you go down a pathway of apoptosis once it's done, you can now actually create two different populations of memory cells. So the central memory cells that stay in the lymph node have these center or clusters of differentiation of what's called 62L. So they express CD62L. This is a CCR membrane receptor 7, and then they make interleukin 7 receptor. So these central memory T cells, they come from an activated T cell that was a naive cell. They stay in the lymph node. The effector T cells become the effector memory cells, and they go out into tissue homing. And in this case, if you look at these markers, they have low expression of 62L, low expression of CCR7, and low expression of interleukin 7R receptor. I'm sorry, high, not low. So low, low, high, 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 high. So these populations stay out in the tissue. This population stays within the lymph node. Okay? So again, they're out there. Second response is faster and stronger than the initial, and you've got a population of lymph memory cells, and then you have a population of tissue circulating cells. Okay? Memory T cells. Questions? Okay, let's try a couple of practice questions. Which of the following is not a correct pairing? CD4 positive helper Ts, CD8 positive MHC1, CD4 positive, MHC2, plasma cell, antibody, APC, fibroblast. How many of you say A? How many of you say B? And C? And D? And E? Okay, the E's would have it. E is correct. So Wednesday's not going to be like we're polling the audience and then everybody writes down the answer, but you get the idea. It'll be fun though, wouldn't it? How about this one? After degranulation, so you have these questions in your lecture slides, but you probably don't have. I don't think you have the answers. Do you have the answers? Okay. Okay, they had to come to lecture. After degranulation, mast cells release prostaglandins and leukotrienes to which to perform which of the following functions? So the question is asking what? What do prostaglandins and leukotrienes do, right? So vasodilation, increased vascular permeability, attraction of CD8 cells, activation of the complement cascade, opsonization of bacteria, or three of the above. How many of you say A? A couple. How many of you say B? How many of you say C? And D? And E? Three of these. And which three do you guys think? A, B, C? So the answer is A. So activation of the complement cascade, how does that happen? That's not prostaglandins and leukotrienes, that's, what's that? Letter. Plasmid, okay, so that you could consider it to be plasmid. I would say earlier on up would be factor 12, which is Hagman factor. So there was a slide in that same series that had the kalkrinin pathway and the coagulation pathway. You guys remember that? Coagulation was on the right side, and then the complement cascade was on the left. Okay? Traction of CD8 cells. They have chemotactic effects, but what cells do they attract? What cells do they attract? What part of the lectures were we in when we talked about leukotrienes and prostaglandins? What's that? Inflammation. And so what's the first cell that you need to attract? So they attract neutrophils. Did we ever talk on Monday or today about CD8 positive T cells being attracted by leukotrienes or prostaglandins? We did not. Make sense? So did you guys remember anything Monday or today about prostaglandins and leukotrienes and their activity on T cells? Okay, so that detail belongs over in the category of inflammation, because that's when we talked about it. So what I'm trying to do is help you guys compartmentalize where the information comes from, because I know it's a lot of information. You're like, I know that's important, I just don't remember where it goes. So if you could think of like categorically when did we talk about it, that might help you eliminate some questions. You guys with me? Okay. Let's see. I think I got one more. Oh no, I got a couple more. Which type of necrosis do you see as a result of glomerulonephritis? Glomerulonephritis. Some of you are like, that sounds bad, but I don't know what it is. Right? It's like nephritis of the glomeruli. Where do you find a glomerulus? In the kidney. So what do you think is happening with a glomerulonephritis that's shown in these pictures? Huh? It's a necrosis, so now is it coagulative, caseous, fat, gangrenous, or liquefaction? Okay. How many say coagulative? How many say caseous? Fat, gangrenous, liquefaction. Okay, I could, that was a pretty straightforward one. Okay, good. That's answer is A, coagulative. Very good. So what you can see is some of these questions you'd be like, oh crap, we didn't cover that, right? That's like your first reaction, like, that sounds like a really complicated question, Keller. We did not cover that. Yeah, we totally did. And you guys got it right. So don't get stung by the difficulty initially of the question. Just break it down. Like, what does it really ask you? Well, I see all the answers. That's why I do like multiple choice for you. Is all the answers are there. The answer is there. You just need to start deciphering which one is extraneous and bogus information and which one is actually real information. Okay. Here's another one. In an experiment, a bacterial organism, I can't even pronounce that, are added to a solution containing leukocytes. Engulfment and phagocytosis of the microbes is observed to occur. Next, a substance is added which enhances engulfment. Which of the following substances is most likely to produce this effect? Complement C3b, glutathione peroxidase, aminoglobulin E, P-selectin, or NADPH oxidase. So break down the question. What is it? There's a bunch of extraneous info here to make it sound more complicated. What's extraneous? Yeah, really the first sentence, right? You can do without it. What is the key? What is the crux of the question? What's the key piece of information that helps you answer the question? Okay, so phagocytosis happens, and then a substance is added which enhances engulfment. What does that translate to, enhancing engulfment? Opsonization. So now we've got something that's going to stimulate opsonization, or more efficient phagocytosis. Okay? Complement C3b. Who says that one? A. How many of you say glutathione peroxidase? Aminoglobulin E. P-selectin. NADPH oxidase. Okay, so that one maybe, this is a good place to study because there wasn't a lot of convincing answers. So it's complement C3b, which helps facilitate opsonization. But you probably want to look at, some of you probably are a little puzzled because, well, all those other ones look important, right? I'll give you one, and then I want you to look out the rest because you're supposed to be studying, right? So P-selectin, how would P-selectin, what does P-selectin do? Where is P-selectin, where did we talk about it? What's that?
Isn't the endothelial cells in the blood vessel?
Yeah, the endothelium in the blood vessel for doing what purpose? What's that?
Sticking to the...
Sticking, so allowing circulating monocytes to roll, right? And then tether and stick and then migrate. What part of the lecture were we on? What topic? Inflammation, okay. So phagocytosis is involved, but it's not at the level of the tissue with phagocytosis. It's happening up in the blood vessel. That's P-selectin, okay. So go through the other ones and see where they are, how they're being used. Help you guys study that way. Okay. Here we go. This is the last one that I have for you, and again, I'll stick around. Yes, sir.
I was just gonna ask, well, we just talked about how optimization is done by antibodies and IgEs and antibody. Is IgE not involved in optimization?
Not typically with IgE because it's IgE's main function is to bind to mast cells and dump histamine. Yeah, so it doesn't have any activity on opsonization or phagocytosis. Yeah. So even like today or Monday, like we never talked about phagocytosis with IgE. You won't see it. Okay. 9-year-old girl sustains a small, half-centimeter long laceration to her right index finger while playing Queen of Swords with a letter opener. Which of the following substances on contact with an injured vascular basement membrane activates both the coagulation sequence and the kinin system as initial response to this injury? Thromboxane, A. Plasmin, B. Platelet activating factor, C. Hagman factor, D. Or histamine, E. How do you say A? B? C? D? Or E? Okay, we actually covered this one in one of the previous questions, right? We talked about, so this is a question about a wound, and it's asking about sequence of events in the wound, and it's talking about the two systems. It even labels them, coagulation and kinin. So it's asking which substances activates coagulation and kinin system. Which one is it? You guys still don't remember? I'm not hearing it. It's factor 12. D, Hagman factor. Okay, do you guys remember which slide it is that I'm talking about? Okay, I'm going to pull it up for you. So that's it. I want to pull up a slide for you guys to see, because it sounds like this one is a source of challenges. I want to refresh your guys' memory. This guy. Okay? Yeah, you're like, yeah, that one. Like, that's one I hate. Okay, so let's just walk through it real quick. Factor 12 is at the top, the Hagman factor. Right? It becomes factor 12A when the basement membrane is exposed. And when you're playing Queen of Swords, or King of Swords, okay, gentlemen. But in my house, it's Queen of Swords. And there's a cut, right? Now the basement membrane is exposed, and collagen on the basement membrane turns this into factor 12A. That stimulates coagulation, and it stimulates the kinin cascade, which also allows for complement to be triggered. And it was correctly said over here earlier, plasmin could have made this happen, but I was asking for something that was further up the tree, okay? On that previous question, yeah?
So on the exam, so say that we had that question on the exam, would you be more likely to put like factor X and Hagman factor together like as one answer? So that's where I got kind of messed up. So if it had
Factor 12, you mean?
Or yeah, sorry, factor
Yeah.
12.
Yeah.
If it had said that, I would have like.
Yeah, probably, I'll try to, or I should say I would have tried to. Exam's done, so I tried to do that. If there are questions about that, like, is there another, you could raise your hand and ask Ryan and I, hey, is there another name for Hagman factor that I might, you know, I don't want, I want to try to get you to answer the question and test your knowledge. We're truly not trying to trip you up. But if it's a question where defining the term is the question, and you ask, you know, hey, could you define this term? Then we'll obviously say, well, I can't really answer that question for you. That's the question. But if it's just clarifying, please ask. It doesn't hurt to ask. Okay? All right, anything else? Can I take a little time? We've got a few more minutes. We've got like 9 more minutes, okay? I actually want to categorically talk a little bit about a couple of things, okay? So let's look at, I want to look at the syllabus. Oh, shoot. We got a few minutes here, so I know you're not late. Okay. Oh, jeez. So this is where we are. And we're right on schedule. And what I want to walk through is each topic. So the first week, we had an introduction, and we got started right into cell injury. And tell me, what are some of the diseases that we talked about? Okay, Barrett's esophagus. That was the first disease we ever talked about together. Okay? What else? What's that? Peptic ulcers. Okay, chronic peptic ulcers. Okay? What else? Yeah, an emphysematous lung from a smoker. We looked at some histology there. Then we moved into cell injury and death, and we compared and contrast two mechanisms: apoptosis and necrosis. Okay? And so tell me some of the characteristics of both apoptosis and necrosis. Programmed cell death is apoptosis. You have blebbing, membrane blebbing. You remember that there was a table, some SEM pictures that were kind of cool. You guys remember those? Which one of those swells? Necrosis, you get cellular swelling initially. Okay, bless you. Nuclear degeneration that's random. And which one? In necrosis. Okay, pretty good. Okay, tell me more about cell injury and death. What else did we cover? Pardon? Oh, the types of necrosis. I think you guys have that down. Doesn't sound like that one's too complicated for you all. You guys don't seem to be missing those. It's those mechanistic slides, those pathways, is where I feel like maybe you guys could brush up by Wednesday. Okay. Then we moved over to inflammation. Tell me what we covered in inflammation. Acute versus chronic. Tell me some of the differences. Neutrophils are the first responders. Pretty good. We've said that like 19 times now. Something like that. I'm not counting, but. Foreign body giant cells. When do those show up?
That's in chronic.
That's in chronic. What else is in chronic? Fibrosis. What is fibrosis? Scarring. Why does it happen? Fibroblasts. It's irreversible damage. Okay, pretty good. We kind of flowed into tissue repair from inflammation because true or false, or there's no true or false, but true or false, inflammation is a key critical step in normal wound healing. True, very true. Okay, good. Tell me about wound healing. We compared and contrast some things. We compared and contrast first and second intention. What else did we compare and contrast? Resolution versus repair. Okay, what are the differences? What's wound resolution? You maintain function, right? Or restore function is probably a better way of saying it. Because you've lost it, then you restore it. Okay. How much scarring is there with resolution? Minimal. Really good. Minimal. We argued over Chainsaw Guy. Somebody thought it was. I know it's not his name, but CG. Chainsaw Guy. Okay, that's pretty good. We talked about some stem cell stuff. Okay. Okay, so let's talk about stem cells. If I say the word totipotent, what does that mean? You almost immediately go to an embryonic stem cell. Okay, I like that. What does it mean, though? It doesn't mean embryonic stem cell, but it can do anything, right? That's like what your fourth grade teacher told you. You can be anything when you grow up. It's like, no, I can't play professional basketball, but thank you for believing in me. Okay. We do have some natural limitations, so. I do not tell my kids, by the way, that they can be anything when they grow up, because you can't. There's certain things you're good at, and there's certain things you're not good at, so. Okay, totipotent. They are told that they can become anything, and they pretty much can, right? That embryonic stem cells is how you get an organism from a single cell. Okay, what's another type of cell? That's a stem cell. Pardon? Pluripotent stem cell. Very good.
And IPC.
IPC. What does it stand for? Induced pluripotent. Very good. All right. And a pluripotent. Give me an example. Some examples of pluripotent stem cells. Not totipotent, but pluripotent. What's that? Adipose derived. That's one. What's another one? Bone marrow. That's another one. Very good. Those are pretty good. How about a placental derived, like an amniotic derived stem cell? Is that considered a pluripotent? Or is it totipotent? It's pluri. It's technically considered an adult derived stem cell because it's not part of the embryo. I know, it's shades of gray, right? Okay. All right, we're winding it down. Then we went into fibrosis and healing. We talked about scarring. We talked about keloids, right? I'm just going off of memory now. And then this week and Wednesday, we were in specific immunity. And that's probably pretty fresh in your mind. So the stuff from Wednesday and today is really fresh. It's that old stuff that you probably should spend a little bit more time taking a peek at, just strategically. Does that make sense? Anybody have any final questions? All right. I will see you all on Wednesday.